guys, today we are going to learn about a macromolecule, and specifically today's focus is about learning about carbohydrates. AP biology exams all have a lot of those questions, and we need to also look at some examples of MCQ questions and see how do they really test you on carbohydrates. So let's go ahead and start. So with carbohydrates, you got to understand that carbohydrates have a carbon. Okay, I'm going to write this down here, a C, a hydrogen, okay, and an oxygen, CHO. And not only carbohydrates are made up of carbon, hydrogen, and oxygen, something unique about carbohydrates is they are in 1 to 2 to 1 ratio. What that means is for every every carbon, you're having a double amount of hydrogen and 1 oxygen. So when we look at a sugar, you'll see that you can count if a carbon has, let's say, like in glucose, like C6. Okay, C6 means it has 6 carbons. There's going to be double amount of hydrogen, H12. There's going to be 6 oxygen, O6, and C6 H12O6 is the chemical formula for a sugar, which is glucose. So it's good to know that. It's also good to recognize sugars as well. If you think about it, sugars will end with OSE. And a lot of sugar names have that. For example, glucose, okay, that's something that ends with OSE. You also have fructose, also ends with OSE. Okay. You have galactose, you have lactose, you have, all of these sugar names will end with OSE. So that's a good thing to know as well. It's also very important to know the building blocks for carbohydrates. When we look at carbohydrates, we see that the smallest, part of the carbohydrate is what we call, that little tiny sugar, a monosaccharide. So I will write that down here, monosaccharide. If you take a monosaccharide, and I like to draw my monosaccharides like this, and if you take a monosaccharide and join it with another monosaccharide, okay, that's when you get something called a disaccharide. And in disaccharides, you will see that, and I'm going to try to draw this. Okay, there's this oxygen here bonded to it like that. And you will see that H2O, water, leaves the reaction, which we call as dehydration synthesis reaction. So to take a monosaccharide and another monosaccharide and join them, you need a dehydration synthesis reaction where water will escape. And if you keep adding more monosaccharides together and create a very big carbohydrate, we would call that a polysaccharide. Okay. For example, a good example would be if you take a glucose and add it to more glucose molecules, you can get a very large polysaccharide. With polysaccharides, we see that we have cellulose, we also have starch, all in plants, and in animals, we have glycogen. Those are some examples. And it's also good to know what they look like. Some AP questions will deal with it. So as we answer some MCQs, we'll go deeper into them and look at them. So let's go ahead and start answering some MCQ questions here. Okay. And look at what we have. Okay, here we go. Here's a question that, and AP exams will do that. The MCQs will have a lot of information given to you. It's about processing and analyzing. Read slowly and let's figure out what's going on here. Here's a question that says different polysaccharides are used by plants for energy storage and structural support. The molecular structures for 2 common polysaccharides are shown in the picture. So here's starch and cellulose, and they're showing you the molecular structure of it, and we'll go through that in a bit. Starch is used by plants for energy storage, and cellulose provides structural support for cell walls. The monomer used to construct both molecules is glucose. So glucose is a monosaccharide, and you're adding a lot of glucose to make this polysaccharide, starch and cellulose, both in plants. A study determined the effect of two different digestive enzymes, A and B, on these two polysaccharides. There was a table of data here that was presented from the study, and what you're looking at here is a table where you have 4 test tubes, and first 2 test tubes, test tube 1 and 2, have cellulose in it, and a third and fourth have starch in it. You also are looking at the effect of 2 digestive enzymes, A and B. So with A, there was no, it says like glucose detected after 5 minutes at 37 degrees Celsius was what they were looking for. And with cellulose, when they added digestive enzyme A, there was no glucose. Okay. With cellulose, when they added digestive enzyme B, though, there was glucose. So looks like it broke that down into glucose. Starch, when you added digestive enzyme A, there was glucose. So that A worked for starch. B did not work for starch. There was no glucose detected. Question goes on. Mammals do not produce digestive enzyme B. So that's B right there. Let me just circle that. Okay. So mammals do not produce digestive enzyme B. So if that's the case, you can already tell, B breaks down cellulose but not starch. So that means mammals cannot break down cellulose because we don't produce B. However, sheep and cattle are 2 types of mammals that contain microorganisms in their digestive tract that produce enzyme B, which makes sense because sheep and cattle do eat a lot of grass and plant-like stuff, and they can digest that just fine. Based on figure 1, which of the following best compares the atomic structures of starch and cellulose? So here's starch and cellulose. Let's look at them, what's going on. If you look at starch, you're looking at a lot of monosaccharides here that formed this polysaccharide. Glucose, here's one glucose, here's another glucose, and they're just held by oxygen right there, and they keep going on. If you look at cellulose, same thing. You have a lot of glucose, but notice what's happening. It's like, here's one glucose, the other glucose is almost like turned. Okay, we call that a 180 degree kind of turn that's happening. Like, think of my hand like this. Okay, that's a 180 degree. Okay, so that's what's happening here, it seems like. So glucose, and then the other glucose is turned like that, and then glucose again, and then that's the difference between the structures here. Let's look at the choices though and see which one of them would explain that or talk about that. So the first one says that starch and cellulose are composed of identical monomers and therefore have identical structures. Well, they are made up of glucose, but they don't have identical structures though. So moving on from that, okay. Starch is composed of monomers that each have a CH2OH group, while cellulose only has a CH2OH group on every other monomer. Also not true. As you can see, CH2OH is something you will see on all of glucose, and both of them are made up of glucose. They'll all have that. Starch is composed of CHNO, which is carbon, hydrogen, oxygen, while cellulose also contains nitrogen. Where did they come up with that? That can't happen because both of them are made up of glucose. Let's read D. Starch and cellulose are composed of repeating glucose monomers. However, in cellulose, every other glucose monomer is rotated 180 degrees. That is the answer, because that's exactly what's happening. Now, based on the same question and the data, they have another one of these questions here. And this one too, we'll have to look at the data table. We'll have to look at the figures. The question here is, which of the following statements best describes the different functions of starch and cellulose in plants? So this is going into functions now. Let's read them one at a time. The monomers of starch are connected by ionic bonds, making it ideal for energy storage. Since I saw ionic bonds right away, I eliminated that because we know it's going to be covalent bonds. Since starch and cellulose are composed of identical monomers, the cellular environment where they are located controls their function. It was starting off just fine, but it didn't end well because it says the environment where they are located is apparently what's controlling their function, and that is not true. Their function has not to do with the environment, but their chemistry at the end of the day. So C says the differences in the assembly and organization of the monomers of these two polymers result in different chemical properties. So there you go. Now it's talking about chemical properties. So I would choose that one. D says the monomers of cellulose are connected by covalent bonds, making it ideal for structural support. Again, it doesn't really explain because it says which of the following statements describe the different functions of starch and cellulose, and it didn't talk about the different functions coming from it. So I would choose C for this one. Now, again, keeping to the same question of cellulose and starch, another one of this one is saying which of the following best describes the process that adds a monosaccharide to an existing polysaccharide. So it will target on a process, and it's talking about adding a monosaccharide. So you have this polysaccharide, just trying to add it. Which describes that the best from the answer choices here? So this one, also with the choices, we have A says that a specific enzyme removes two hydroxides, one from the monosaccharide and one from the polysaccharide. Now, we do need enzymes to do that kind of stuff, but again, this doesn't quite best describe the process that's adding, though, a monosaccharide to existing polysaccharide. So I think there should be a better one in here. Let's keep reading. And it says creating a bond between the two monosaccharides and creating a H2O2, hydrogen peroxide molecule. Absolutely not true. B says a specific enzyme removes the hydrogen from the monosaccharide. Okay. And we know that can happen. And a hydroxide from the polysaccharide, creating a bond between the two and creating a water molecule. That kind of sounds like dehydration synthesis reaction. So if you remember, you have hydrogen from one and OH from the other one, and they do release water. So that does sound like something that would be a good answer. But let's keep reading. Ionic bonds? No, I'm going to move on from C. The monosaccharide is completely broken down by a specific enzyme, and then the atoms are reorganized and made. That's just so wrong. So really, B was the right choice here, and we'll go with B. Sticking to the same type of data question we were looking at, this one is asking which of the following would most likely occur if cattle lost the ability to maintain a colony of microorganisms in their digestive tract. Remember, the reason cattle can't digest cellulose is because they have those microbes that have the enzyme to break down cellulose, because it's not easy to. Mammals, we don't have it. So this is a good question, because it's saying which would happen if they lost the ability to have those bacteria or microbes. Okay. First one says cattle would have to start producing enzyme. We know that's not going to happen. You don't just start producing an enzyme. You either have it or you don't have it. You don't just start producing it. That's not going to happen. You need the gene for it. So not going to happen. Moving on. Cattle would no longer be able to use cellulose as a primary source of glucose. That's kind of true, because if you don't have the enzyme, if you don't have the bacteria, they would not actually be able to use cellulose and break cellulose down to extract the glucose to make their energy. So that kind of sounds like something I would pick, but let's keep reading. Cattle would have to convert cellulose to starch. They can't do that. So again, not going to choose that. Cattle would no longer be able to synthesize. They've never synthesized cellulose. They eat it and hopefully break it down with the help of bacteria, but they never synthesized it. So really B was the right answer choice for this one. So that's kind of it. This shows you what kind of questions can come from carbohydrates, but it's really important to understand the structural parts, the chemistry, the monosaccharides, the dehydration synthesis reaction, in order for it to answer questions like this. All right, we'll learn about another macromolecule next, but this is all for today.